Hello everyone and welcome to today's webinar, Flow Cytometric Gene Mutation Assay Made Possible by Immunomagnetic Separation Achieves OECD Regulatory Status. I am Cassie Saltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Jeffrey Bemis, Director of Clinical Studies, Litron Laboratories, and Joe Ostrand, Flow Cytometry Specialist with Miltenyi Biotech. Jeffrey, you may now begin your presentation.
Okay, thank you very much for that introduction, and we'll just jump into these slides. So I'm going to start out, if you're not familiar with us, with a quick introduction to Litron. We've been doing genetic toxicology for over 40 years. Primarily, our approach is in the development of new assays and novel methodologies. The bulk of that is really done in preclinical product safety, but we do have some clinical applications for our methods as well. As you'll see throughout the course of this talk, we are very flow cytometry centric. So all of our assays to date have been produced to use flow cytometric instrumentation. The broad theme for today is that automated assays and instruments are going to yield better information for you in your studies. This can come out as improved compound characterization. They can be a complement or they can achieve regulatory required assays. And I'll give you an example of that later. It can greatly expand the range of information or the type of data that you're going to be generating from your studies. And that can help you make decisions about what to move forward and what might be a hazard for you. And then finally, they can address many of the 3 R's initiatives, and I'm sure you're familiar with these, and it makes a lot of sense when I start talking about in vitro assays. But even in the case of the in vivo assay that I'll be describing today, you can make better use of the animals that you have to use, or they're going to be in your required assays, because of our ability to just use a small amount of blood and get multiple endpoints out of those animals that are already used for the conduct of your regular studies. Specifically, as the title said, we're going to be talking about gene mutation assessment in red blood cells. Now, this is commercially marketed as our in vivo Mutaflow assay, and I'm going to start out with a basic introduction to what that assay is. If you're familiar with this assay at all, you may, and you probably have heard it referred to as the PIG-A assay. That PIG-A comes from the gene that we take advantage of to create this assay, and that's the phosphatidylinositol glycan complementation group A gene, or the PIG-A gene. It's specific and is critical in the formation of GPI anchors. And these anchors are structures that are used to attach specific types of proteins to many different types of cells. In this case, for this assay, we're going to be talking about the surface of red blood cells. This gene is also causal in the human disease paroxysmal nocturnal hemoglobinuria, or PNH. And much of the biology that we understand about the assay that I'll be describing today has actually come from research into that particular human affliction. The other characteristic that makes this critical, the PIG-A gene, an advantageous target for this assay, is that it is located on the X chromosome. So that means whether you're an XX female and one of those X chromosomes is silenced, or you're an XY male and therefore you only have one X chromosome, it only takes one hit to kind of produce this cell surface phenotype that I'm going to describe and show to you on the next slide. So it's really more of a rare event type of analysis as opposed to, say, an autosomal, expressed chromosome or an expressed gene where you'd have 2 or more copies per cell, and therefore to knock both of those out, you would have to have it would be a very, very extremely rare event. So in this case, we're kind of taking advantage of the one copy of the PIG-A gene per cell, and therefore we can reduce that down to assessment of a phenotype that can be reliably assessed by a standard flow cytometric, antibody cell labeling type of process. And what that looks like here is kind of shown on this slide here. We're doing this in red blood cells, as I described before, so I'm taking that specifically here. And on the left-hand side of the slide, we have that structure. We have the plasma membrane that you can see here in the small rectangle. Inserted into that is the GPI anchor. So there's that structure that is used to tag the CD59, which is that protein that we're showing here as a GPI anchored protein on the surface of those red blood cells. And it's kind of simple here. On the right-hand side, the mutant phenotype, none of that is there. That cell could be expressing all of the CD59 that it possibly could, but it will never be trafficked and adhered to the surface of those red blood cells because the GPI anchor wasn't formed, because the PIGA gene wasn't functional in that particular mutant cell. This is just a quick diagram of that to show you this is the phenotype that we are assessing by flow cytometry. We move back over to the left-hand slide, or left-hand side of the slide. We incubate the cells with a standard fluorescently conjugated antibody against that CD59 and run a standard flow cytometric assay as you would for any type of these cell surface markers. Well, what do we actually get out of that when we run that? What kinds of output do we get? Well, starting over on the left-hand side of the slide, again, we were looking at red blood cells. And in fact, we look at two different types of red blood cells. We look at reticulocytes, which are the less mature, the red blood cells that are just coming out of the bone marrow. And that is considered our leading indicator of mutation here. So if we look at the plot here, or the line graph, down along the bottom, we have days of exposure to ENU, which is a well-known, well-established mutagenic agent. This is in Wistar rats. And then we're looking at the average number of CD59 negative. So these are the mutant reticulocytes that we're looking at here on the y-axis. And you can see here by about, day 10 or so, we're starting to see elevations in those mutant reticulocytes at different concentrations. It's a dose response in a time-related fashion. We're seeing increased mutation frequency in the reticulocytes, or mutant output there. We move over to the middle of the slide, and we're looking at the more mature erythrocytes. So this is the bulk of the red blood cells that exist in the periphery. And as you can imagine, there are so many of those cells there. It takes quite a bit of time for those mutants to, say, turn over and to actually sort of become frequent enough in that population to be detected. And therefore, you can see we're starting to see significant elevations right out at about 28 days to 48 days or so. That's where we really begin to see the major impact on that much larger, more mature red blood cell population. So we sort of have these two indicators, this leading indicator and lagging indicator of mutation within the red blood cells. We also get information on just the frequency of the reticulocytes themselves. So not the mutant reticulocytes per se, but the combination of the mutants plus the wild types. And this isn't necessarily so much an indicator of the mutation that may be happening in the system, but more it's a reflection of sort of toxicity in the bone marrow. And in fact, it can help you determine and in fact prove that you've actually gotten your test article that you gave to the test system. You've actually targeted it and gotten it to the bone marrow, and you're having some sort of an effect in a depreciation of reticulocytes. A toxic effect in the bone marrow is being reflected here just in the frequency of reticulocytes in general. So those are the three standard parameters that we get out of our assay. So it seems pretty straightforward, and it sounds like just a standard antibody, combine it with cells type of assay that you would be doing by flow cytometry that most flow cytometrists would be familiar with. But as I'll show you here on the next couple of slides, we were presented with several methodological challenges that we had to overcome, and the biggest one of those is reflected here. This is sort of what we're calling the basic setup, the assay that when we started it out and began the initial development of this assay, we ran into this complicating factor. And you can see it here because the number of red blood cells that we were looking at is around 1 million per cell or per sample. So per blood sample that we took from an individual animal, we were getting about 1 million red blood cells. Those are the mature erythrocytes that we were looking at. We were getting about 0.3 times 10 to the 6 of the reticulocytes. That's a much less populous portion of the red blood cells in the periphery. Therefore, as one would expect, we're counting a lot less of those. It would take us about 20 minutes per sample to do this analysis, to get that number of red blood cells and reticulocytes. Probably, kind of long for many people to sit there in front of the flow for a single sample. But, in this case, we were hoping to get some useful information out of that. Ultimately, this resulted in relatively low accuracy and precision putting those values on there because, as you can see here, there was a very low spontaneous frequency of PIG-A mutation as a baseline. And as you can imagine here, the baseline was right around 1 per million cells. So if you can see that we're trying to develop that and accurately score that value, that frequency in those reticulocytes, but we're scoring less than 1 million, you can imagine we saw a lot of zeros in that. And that's really hard to actually put numbers on and run statistics on if a significant number of zeros are coming through in your system. This just really wasn't making this a feasible approach to accurately and reliably quantify the populations that I was just describing on that previous slide. So to overcome that issue I was just describing, we are going to employ the Miltenyi superparamagnetic columns, and we're going to enrich for the particular population that we're interested in, just like that technology is used in any number of other applications. I'm going to show you specifically how we do that and then combine it with the flow cytometric technology that Miltenyi presents in their instrument line of flow cytometers. So here you can see a relatively complicated slide, but I'm going to reduce it really down to a pretty simple concept in that we label the wild type cells, the bulk of the population of those red blood cells and reticulocytes that are in the blood sample, with an anti-PE microbead. Those are the superparamagnetic, or those are the beads that get used in combination with those columns to then hold up that population on the column. So we're really reducing the vast majority of cells. We're holding them up on the column, and we're eluting out the population that we're really interested in focusing on, the wild type cells. A few of the wild type cells do come through, excuse me, but all of the mutant cells, the mutant reticulocytes and the mutant red blood cells come through, and then we can label them again with a bead solution that allows us to enumerate the wild type cells that are there, the mutant cells that are there, the beads that are there, and come up with a mutant red blood cell to bead ratio, a mutant reticulocyte to bead ratio, and then compare that to a sample that we took prior to the column, do a little bit of mathematics, and ultimately come up with the indication of the mutant cell population in that entire blood sample. So we virtually score a significant increase in the number of cells that we can run through the flow cytometer. And I'll show you the math on that in a little bit. What it really looks like on the flow cytometer is shown here. Over on the left-hand side of the slide, this is our pre-column sample. So we took a sample of the blood before we ran it through the column. It's already been labeled with all the antibodies and beads that are required for the column separation. And you can see here on the right-hand side of this plot, the significantly large population of the wild type cells. So that's the predominant population that's there. That's the one that, in a sense, is sort of clouding all of the assessment of the mutant population that is over on the left-hand side in that red circle. You can see there are some of them there, but it's quite difficult to actually put a significant or a good number on that that's reliable when we're actually looking for a good number on that population. So this happens to be an ENU treated animal. So there's a significantly large number of those, that cell population there. So that's why you're actually seeing them show up in this population. When we move over to the post column, where again, we've held up the vast majority of those wild type cells, you can still see some of them are there. And in fact, we want some of them there so we can use them to position our quadrants appropriately. But we can focus much more reliably and efficiently on the left-hand side of that plot and look at the mutant populations of the reticulocytes and the red blood cells. And as you can see there, it's much easier and more efficient for us to put a reliable number on that population. Ultimately, how does this compare to that basic method that we were describing before? This is the same portion of the slide that I was showing previously. We get that, million red blood cells, significantly less than 1 million reticulocytes. Now with the Mutaflow system, we can count over 100 million of these red blood cells, 3 million reticulocytes per sample. We can do it significantly faster. So we're not on the flow cytometer for 20 minutes per sample. It's around, 4 minutes per sample, much more reasonable time input on the machine. And as you can imagine, this results in significantly higher accuracy and precision associated with the numbers that we can achieve with this new approach using the Miltenyi columns. This then really allowed us to reduce this assay to practice and get it out there in commerce in the form of our kits and our services. So this was really what made it possible for us to develop a viable assay for the assessment of mutational damage in an animal system. Speaking of those kits specifically, well, how do maybe it's just Litron that's sort of really good at this. How do we sort of prove that other people can sort of take these kits on and use them? We do an awful lot of these transferability demonstrations. And in this case, and we're going to look over at the left-hand part of the slide first, we did an experiment with some colleagues from GSK and Covance, and we sent them the kits. We all exposed animals to either untreated controls or that ENU compound, again, a well-established mutagen that was going to cause significant elevations in those two populations, the mutant reticulocytes and the mutant red blood cells. In this case, we're actually looking at the CD59 negative mutant red blood cells. You can see here, very great, very nice agreement between the different groups. So we were all looking at our baseline controls there in the untreated controls, and we're looking at significant elevations, but very consistent and reliable numbers that we were able to put on those elevated values due to the ENU exposure across those different laboratories, again, using all of the same reagents that came from Litron. We dig a little deeper into the data, and we can look, in this case, specifically just at Litron's values and those from GSK. What we're doing in this case is we took the same bloods that we were looking at in that previous plot, and we mixed them together at various dilutions. And we're looking at an expected CD59 count versus what we actually measured. And you can see here excellent correlations between the expected versus the measured values, great R correlation coefficients at both Litron and at GSK. We took it a little bit further and we did a Bland-Altman analysis, and we can see here, very low bias, very narrow and consistent and reliable accuracy and agreement within those methods across the different groups. So really a great assessment of performance in the transferability or the ability for other laboratories to conduct this assay. So we've got the assay, we've got it together. Well, what do people actually do with this? And how does it sort of get out there into the wild and start having an impact on the kind of work that we're doing here? I'm going to talk about that in the context of these sort of regulatory challenges, because ultimately the best place or the greatest impact that we've had with this assay has been in sort of these regulatory safety studies that are being done for, pharmaceuticals, industrial chemicals, et cetera, for their safety submissions. And therefore, once it gets through the sort of regulatory framework and can work within that regulatory framework, it'll probably get much greater acceptance. So on this part of the talk, I'm going to sort of describe the saga that we went through in order to get this as a OECD, worldwide test guideline on how to conduct the PIG-A assay. So starting all the way back really at the beginning of where I was talking about the development of the assay, and we were doing those interlaboratory trials. We looked at, upwards of 70 different chemicals across multiple different laboratories. It took us a number of years. You can see at least 5 years we spent on doing those types of interlaboratory trials. And in the middle of that, we had to sort of gather back again and refigure that out and reconfigure it for use with the column technology. Ultimately, we got that together and we got it working very well. And so we proceeded forward. We were looking at the assessment and performance of the assay in the context of several of these IWGT and HESI, GTTC. They're basically just expert working groups that all came together to look at the data, to look at the different, performance assessments that had been done across the different laboratories and really judge whether or not this thing was, again, worthy of moving forward into an OECD guideline. Ultimately, it was judged to be quite successful. And then we started going through the process of submitting an SPSF in 2014, getting a work group together in 2015. These are all under the guidance of the OECD framework. The detailed review paper was submitted to the OECD in 2017. Ultimately, an expert working group was formed within the OECD to comment and to create the wording that goes into the guideline. The detailed review paper, a retrospective performance analysis were all approved in 2018. The drafts of the exact guideline were circulated around 20 to 21 and revised and commented on by all the different stakeholding countries that are in the OECD. And then ultimately, this was accepted as a OECD approved test guidance in 2022. So very recent acceptance for this assay. Here it is right here. Here's the OECD 470 test guideline. This is a picture of the cover of it, and you can see the title Mammalian Erythrocyte Pig A Gene Mutation Assay. Ultimately, what this does is it describes the use of these methods, which includes the multi-flow assay, which is commercially available and was the one that was developed at Litron, in how it gets used as part of regulatory safety studies. This serves really as a guideline primarily for pharma and chemical companies in their industrial uses. You can use it as part of standard product development. So if you have an issue with, say, you got an Ames positive or some sort of indication of mutation in some of your in vitro assays, this would be a great follow-up to that. One of the places where it served greatly out in the pharma community, especially most recently, has been in the context of impurities in the production of their pharmaceuticals. If you're familiar with this issue that has kind of been plaguing pharma for a number of years, the steps in some of the examples that I'm showing here talk about the inclusion of the Pig-A assay as a useful tool was laid out in the ICH M7 guidance, which is sort of a guidance for pharma on how to deal with impurities and how to manage those types of things in the product, in the development of their products. It was used quite effectively in this pharma consortium of number of different pharmaceutical companies that came together to look at the impact of boronic acids on some of their synthetic processes. And then very recently, you may have been aware of the impact of nitrosamines as these mutagenic compounds that get formed in the synthesis of certain types of pharmaceuticals and can be impurities and can be contaminants in certain pharmaceuticals, and they need to be controlled at very specific levels, and we need to understand whether there is a hazard associated with those. And again, the Pig-A assay had played a great role in that particular exercise. So to conclude, our in vivo mutaflow assay is able to deliver mutation assessment in red blood cells and reticulocytes in an in vivo system to give you that gene mutation endpoint that you may be looking for your regulatory safety studies. This is easily accomplished by flow cytometry, as I described to you, now that we have that mutaflow system that incorporates the scoring of a sufficient number of blood cells in each of those populations. Key to that and critical to that was that column-based isolation that I described, and it is going to be talked about a little bit later in this presentation. A couple of other endpoints specific to this mutaflow assay is the integration of this assay into ongoing toxicology studies. I mentioned it way back at the beginning of this, that small amount of blood that you need to assess this endpoint really makes it easy for you to sort of drop into, say, a 28-day repeat dose toxicology study and get this in addition to all of the other endpoints that you're looking for. So you don't have to do dedicated genotoxicity studies like you might have used to in the past. And we've developed it for primarily the standard laboratory, mammalian species that you're going to be looking at, rat and mouse, and a couple of other species as well. So what are the takeaways from my part of the assessment here, the talk? This combination of advanced assays and state-of-the-art technologies is obviously quite powerful. It was critical that we use that column-based enrichment to achieve the numbers that we were looking for, and then you can synergize that with the high-throughput instrumentation that Miltenyi and other flow cytometry instruments have, and that was really the key to delivering a viable and reliable assay for the assessment of mutation in vivo. This mutaflow assay has ultimately turned out to be a highly accurate and reliable gene mutation assessment methodology. It is available in kits that are that come with our QC reagents, that come with a significant amount of technical support. We've also have it as a service, so that if you don't have the flow cytometry or the expertise to do that kind of thing in-house, you can contract with us or a number of other CROs that can conduct the assay and actually get those values for you if you aren't able to take receipt of the kits and do it yourself. Yeah, so I just want to acknowledge a couple people at Litron that have been instrumental in the development of practically all the assays at Litron, but primarily the Pig-A assay that we were talking about earlier. Svetlana Oblasevich, Dorothea Torres, Steve Bryce, and Steve Dertinger are really the main people who have contributed significant amounts of time and effort to develop this assay. And this was, I also want to acknowledge that this was done in the context of our SBIR funding that came from the NIEHS. Several different grants contributed to, over the years, the development of this Pig-A assay. and to really reduce it down to the functional system that we have available today.
Thank you, Jeffrey, for your presentation. We're now going to pass things over to Joe Ostrand, our next presenter. Joe, you may now begin your portion of the presentation.
All right. Thanks for that awesome introduction, Jeff, and really cool depth into that assay as well. My name's Joe. I'm from Miltenyi. We've actually provided a lot of the equipment to help support this assay, and some of the core technologies at Miltenyi are critical to making this assay successful. And I'm going to be taking a few moments to describe one, the magnetic enrichment technology that allowed for selection of cells, and then two, some details on our cytometer, the MaxQuant 10, and how that helps support the GeneTox workflows, as well as many cell-based assays. I've been with Miltenyi for about 5 years now on both the technical team and now as a senior cytometry specialist. So I feel like I understand these technologies quite well. And yeah, we'll have some time at the end for questions, but I'm basically going to go over some of Miltenyi's main technologies and hopefully in a little bit of detail, so that even if you've used them before, you may learn something new. And for those of you completely new to Miltenyi, I hope this is a good background on what we do. So to start with, Miltenyi's a company that really works in the translational space. So we have a variety of technologies, both on the reagent side and the instrumentation side, that help researchers develop technologies, develop assays, and then transition them to a regulated environment. And so our sort of main slogan for that is from benchtop to bedside. So we really pride ourselves on our ability to transition technologies and transition assays to this regulated environment. And we have a huge support team to help make that possible. If you're working with cells, if you're working with tissues, these slides are meant to show that we can go from something as big as a tissue, break it down using the GentleMax, which is our instrument in the top left, work through a variety of cell preparation strategies, whether that's isolation or activation, maybe culturing, and then going through and looking at those cells and seeing how they've changed in that process. For the purpose of this discussion, I'm not going to get into every single technology or every single piece of equipment, but I am going to be focusing primarily on the magnetic activated cell sorting technology. This is, if you've heard of Miltenyi, you know us for this technology. And like I mentioned, I'll also be talking about the cytometer because that's really my area of expertise. So starting first with the magnetic technology, this was really what Miltenyi was founded on. So Stefan Miltenyi developed this technology in 1989, or really marketed this technology, created an industry around it. And effectively what it is a magnetic particle attached to an antibody that would be specific for a cell type of interest. So that allows you to attach a magnetic particle to a cell and then use a magnet to help enrich or deplete that specific cell out of your starting material. So this has been influential, really field changing with respect to your ability to work on individual cells or individual types of cells. And it's also created a whole clinical market as well. And really beyond just a clinical market, it's created clinical therapies that have helped people with a variety of disease states that you need to enrich for cells for. So I think it's been really remarkable to be a part of Miltenyi, to see how this field has grown. Just with even in the last 5 years, I think Miltenyi's probably doubled in size since I joined, simply because of how profound and how simple it is to work with these microparticles. I'm going to go into a little bit of detail on how these things work and why they're particularly helpful in this context. But in order for them to work, in order for Miltenyi's version of this to work well, we have kind of three main components. We have the microbeads, which I've just described, the magnetic particle with the antibody attached. We have a column, which you will run that now magnetically bound cell population through. I'll go into some detail about how those columns work. And then the separator, which is the actual big magnet that we use to help generate a magnetic field and make that separation occur. So moving on to the next slide here, we've got a zoom in on these microbeads. So the microbeads are 50 nanometers. They stay in suspension, which is what colloidal means. It's nice they don't like drop out in your tube. I know I've using these in the past, I always shook them up to make sure they were resuspended, but I've since learned since joining Miltenyi that you actually don't need to shake them up. They're super small. So this is the major difference between us and maybe any other magnetic microbead particle technology you've used. Ours are incredibly small. So that means, one, we don't have to bind up every single antigen on the outside of your cells. And two, it means that they don't necessarily activate the cells as readily as some of those bigger particles. So in that regard, your cells are more useful for downstream assays. So if you're not binding every antigen, that means some antigens are available for binding flow cytometry assays or binding flow cytometry antibodies too with fluorescent dyes. It also, because they're so small, they're actually able to be digested by the cells too. So at some point they're internalized and actually broken down by many different cell types. So in the regulatory sense, those particles are actually not a concern to be a part of your final clinical product, which is really interesting. So as stated here, they're non-toxic, they're biodegradable and biocompatible. So I think these all make for a very translational technology, something that's really high utility across the field. Now, in order for the microbeads to work, because they're so small, we use a column to help encourage a stronger magnetic field to still get the same amount of separation pull without having these large beads, which affect cell livelihood and viability. So in the simplest sense, we label the cells with the magnetic particle, we do the separation via a column, and then you elute out your cells of interest. There are a couple ways that this can happen, either via depletion or separation, but they all take advantage of this column magnetic force growth. So we're using magnetic particles in these columns, and the columns themselves simply expand that magnetic force. So by using the column, you're able to use these smaller magnetic particles, which have a lot of advantages. The columns have plenty of room in them for cells to go through. Sometimes people are curious about if we're losing cells in the process of even just going through the column. But for the most part, the space between the beads that are packed in the column is plenty for cells to move through, which is important because we really need cells to move through the column freely in order for this to work well. So we've got a couple of quick examples to show positive enrichment and negative enrichment, sometimes called depletion. And so these two are really the core ways that we use these columns to enrich for cells effectively. So this little diagram will show positive selection. In this case, we've labeled the cells that we want. So in the diagram, you're going to be looking for the green cells. The green ones are the ones that we want in this graphic. And they've been bound with the magnetic particle. And so when the magnet's being applied, the cells that you want stick in the column and everything else washes through. So this is really nice for a really clean, high purity enrichment. And the caveat, of course, is that you've used an antibody to bind to your cells. I think in many cases, this binding is not a big deal to the cells of interest, but some people like their stuff with nothing bound to it for a variety of reasons. So again, this is showing just the positive selection binding to the column itself. The other way we can do this is called the negative isolation, sometimes, like I said, called the depletion. In this case, we're looking for the purple cells. Sorry, I think we're looking for the purple cells in this one. And we've used antibodies to bind up every other cell type that's in there. And those other cell types will stick in the column, and the cells we want are going to process through to the bottom. And that elution is what we'll take away. So in this case, we've got a really nice clean or untouched population that we've enriched for. The tricky part for this one is that you need to make sure you have an antibody for every single other cell that's in there. So if you don't account for some specific cell type in your population, it may elute out with your cells of interest. So these are all things we've thought about all the way through in the development of these products. So this is not a surprise. And certainly our pre-built kits will absolutely have every antigen you need to bind up cells that are not wanted in this regard. These have a bit more difficulty or complication to go into a translational market because of how many different reagents or antibodies are involved with the depletion, but that's certainly something that we could talk about if anybody's interested in making that transition. So all of these technologies that utilize the column, it is the core technology for Miltenyi. So as you may imagine, we have a variety of formats that these come in. And these help researchers support either very small scale or entry level research-based enrichments all the way up through huge, taking down big blood products from patients or donors and doing this in a really high throughput format. We also have technologies that make things either more automated or simpler to use or with fewer total reagents. So overall, there's pick your flavor, right? So no matter what you're working on, we may have something that's really tuned to your need. So starting on the left, we have the low throughput. This is just a plastic column that you would run through just a single sample. The one to the left is basically 4x that. I think there's even one that's, you can take 4 columns and stick them in there. The columns come in a variety of sizes too, based on your needs. In the middle is the Automax. This is an automated system that will process and spit your cells out for you without really any hands-on time, which is super nice. Next to that is the Multimax 24, which allows you to basically multiplex, do a lot of these enrichments at the same time and process through up to, I believe, as high as a leukopack in a single process, which is really convenient if you're trying to do large-scale enrichments. And then at the extreme side, we have a fully automated system that will do liquid handling and processing of large blood products and spit out a huge variety of things. So these things have, really anywhere you fit in this scale, there's something for you. And it may be very, very easy to work with as well. So moving on from the magnetic technology, which I think we've described in plenty of detail, the assay that the pig assay that we talked about utilizes cytometry in a gene tox setting, which is a nice transition. Cytometry is really great for this world, and it's something that it's been exciting to see adopted by more and more of these technologies. It's just you get so much data so quickly with the cytometer, and I really think it's a perfect technology for this type of work. So we're going to talk a little bit about our cytometer, the Max Quant. We have a variety of types of cytometers that fall under the Max Quant designation, but we'll talk about our cytometer and how they fit well into this environment. So I mentioned that gene tox, or really toxicology in general, is starting to adopt cytometry over microscopy, and there's a few very important reasons why that is. I mentioned it's faster, so you can process thousands of events in a single second. This is, by microscopy, that's incredibly challenging. And oftentimes a cytometer can give you a readout that is easier to comprehend to someone that's maybe not an expert. So you put it in this a cytometry gate, it can spit out yes or no. Whereas microscopy, there can be some interpretation that is needed by somebody who's really got an expertise for this. Cytometry can be automated in a much simpler way than microscopy, and you can also automate the processing of multiple samples, which is really nice. So outside of cytometry as a whole, the Max Quant specifically has a couple of elements to it that I think are very nice for this environment. So first, because it's based on a syringe, we have absolute cell counting, which means you can put your sample on there and it's going to spit out not only the frequencies of each of those populations, or the plus minus, positive versus negative, but it's also going to tell you exactly how many cells and the concentration of those cells in your material. So if you're looking at a killing assay and you need to say how many cells are left over, if you're looking at really anything that requires you to quantify the number of cells in there, you can do that very simply. So we've written on the slide, it saves money, which is of course true if you're not buying counting beads, but counting beads are also a huge pain to work with. So if you're able to remove those from your assay, then that's really advantageous. The MaxQuant also has a very small dead volume. So a dead volume is additional material that needs to be put into the cytometer in order to measure your sample correctly. So let's say if you have a standard cytometer and you're measuring 20 microliters of your material, you may need to add up to 50 microliters more in order to measure that effectively. And that number can go very high depending on how much automation you're using. Whereas the MaxQuant, we're much lower than that. So in any case, this is very nice if you have limited amount of sample. MaxQuant also has onboard washing, so that minimizes carryover from one sample to the next. So if you are looking at very, very rare events, or the presence of an event is particularly impactful to your ability to either release product or qualify your material in a QC setting, the carryover can be it can make your assay a lot less reliable if your carryover is not accounted for. So with the MaxQuant, you can automate a cleaning cycle that brings our carryover down to 0.01%, which is far lower than anything else on the market. We've also worked with Litron for quite a long time to develop automated templates that work with Litron kits. So this is overall, like if we're talking about technology on the cytometry side, it's all great, but what you end up really, really wanting is a partnership with the assay kit builder and the technology or the instrument builder. And so the fact that we've worked with Litron so closely throughout this process is incredibly valuable to the end user, because if you're troubleshooting problems, a lot of times if the companies don't communicate, if they're not collaborating, you get sort of a finger pointing thing where you're not sure if it's the kit's fault or the instrument's fault. And because they're not working together, they may not give you a great response. But both the Litron team and the Miltenyi team have a great understanding of both of our portfolios, to the point where we can give really, really detailed support on any of the assays that you're using between our companies, which is really great. And I've worked with a handful of folks over at Litron, and it's always been a really easy thing to work with. So definitely this is probably my favorite part about our technology in this space. So I mentioned we have a few MaxQuants. We have the MaxQuant 10, which is our kind of our flagship cytometer. This is something that I think is really robust in the QC environment, something that is very easy to use for new cytometry people, and basically checks off all the boxes that I've just mentioned. Also in our scope is the MaxQuant X. This is a high throughput version that basically runs 384 well plates as well. So we can accommodate whatever scale you need. On the analyzer as well, they're good systems. We've got good cell counting. They're easy to work with, easy to maintain. And we have a couple versions with respect to number of channels you might need. So if you're doing something like immunophenotyping, we may have more channels for you than somebody just doing a one color toxicology assay. Everything's run off of a pipetter needle here, which is fairly different from any of the other cytometers on the market. This one allows for really good volume characterization, but also lets us do mixing in the plate. It lets us switch between plates and tubes really easily. So any of that stuff is very straightforward to use. If you are using tubes, we have a rack that is chilled. So you can put it in the fridge and keep your samples at a cool temperature while you're operating. So this can be really helpful if your assay is very temperature dependent. And we have a reagent rack here, which you'll see on the bottom right, which allows you to actually automate some of the either antibody additions or a viability dye addition, which is super nice. If you're in this setting, it's often that you're developing an assay that's going to be done over and over and over again. So some of these really simple automation strategies can speed up your assay or remove some of the requirement for an additional headcount to do this kind of thing. All right, I'm going to bring up all these at once because I think I don't necessarily want to read every single one, but Miltenyi's done a lot in this space. There's cell-based assays are a huge part of what our magnetic enrichment is used for. And so a lot of our cytometry, therefore, is designed to support these assays as well. We have screening assays, immunogenicity assays, which I think are becoming more and more popular by flow. Toxicology screening is, of course, important. This is probably why a lot of you are on the call to begin with. This micronucleus test is one that we have many cytometers in the field to support. So we really run the gamut on these cell-based assays. I find that in general, you're not using a huge amount of colors, so you don't need a crazy 5 laser system, but you need something that's going to work for you and continue to work for you over a period of time. So if you see assays on here that you are using and you want to get in touch with us, absolutely let us know. If there's other assays that you're not sure if you can put into flow, let us know too. All these came from somewhere, right? So we have application development scientists that can work with you to take something that's maybe a microscopy assay now that you want to automate. Those are all fun projects for us, so definitely let us know. So finally, we've got a small cytometer. It's got all the lasers you would need for most toxicology assays. We have lots of different ways to run tubes. It's easy to operate. And one thing that I haven't touched on is we have really integrated support. So that's live support where we will either be on site working with you to do application support, but we also have a button that you can click in the software that will connect you to our tech support team, and they can actually look at your screen. If you give them permission, they can click around on your screen and help diagnose any challenges you're having and actually help build out some of the data set you're looking for too. So all that stuff is very simple to connect with, and something that's really quite unique to our system as well. So with that, I'll wrap up on the Miltenyi side. I have a slide up here to sort of thank all the folks over at Litron and my colleagues as well at Miltenyi. And I think our teams have worked really nicely together. As you can tell, we're doing a webinar together, but the amount of stuff that Litron has done with us is quite big at this point. So we're really happy to be connected with them and to continue doing work like this. And yeah, we're happy to start taking any questions now. And we'd love to hear from you guys. So thank you.
Thank you, Jeffrey and Joe, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, can the Pig-A assay be incorporated as part of a repeat dose tox study?
Yeah, so the short answer for that is yes. I mentioned it very briefly towards the end of my presentation, and I was specifically talking about, say, a 28-day toxicology study, which, if you're familiar with that, then it's going to sound like, something that is fairly standard conduct for safety assessment of pharmaceuticals and industrial chemicals, et cetera. But to go into a little bit more detail as to how we actually fit it in there, you heard me talk about a low blood volume requirement. For this Pig-A assay specifically, you really only need a maximum, it's somewhat species dependent, but about 300 microliters of blood. So it's not a very large ask in terms of the volume of blood that you would need to go towards this. So that's fairly simple for you to get from numerous routes in rat or mouse. The other component of this that I didn't get a chance to address is you can take these samples and you can process them fresh. If you're running them right in your laboratory, you're going to collect them and then run them the same day. You can keep the samples fresh and you can run them immediately or within several days. So they're good for about 5 days if you want to keep them refrigerated, maybe get to them the next day after your collection. But we also have a means for freezing samples, and this is really of great advantage for those tox studies because maybe you're conducting this study for numerous other reasons. You're getting histopathology, you're getting TK, PK, all those different other components that you generally get as toxicology studies. And maybe one of those is the contributing factor to the dropping of that particular compound. You blew out the liver or something like that. If you froze our samples away for, say, PIG-A or mutational assessment, you don't have to run them right away. You can store them for months at a time and get back to them. And that way, if it comes up that you needed to look at a mutation endpoint later on, you don't have to start a whole other dedicated study and invest in all the time and the resources to do that again. You simply dip into the freezer where you froze those samples away, and you can get that mutation endpoint. So there's several characteristics of this assay that really drive it home towards that 28-day toxicology study. And in fact, that was really the study design that we used for the development of the assay. So the bulk of the data that are out there and that you'll read in the literature were based on 28-day toxicology studies. So it really is well established for that paradigm.
Great, thank you. Our next question is, what species are compatible with the PIG-A assay?
Sure. I mentioned it again right towards the end of my presentation. I think I said rodent studies. We have kits and services specific for rat. That's probably the most common test system that we work in. And there's a slightly different, there's a slight antibody change to go over to mouse. So we can support both of those very common laboratory-based species. And then the other system that we do have an assay developed for is human. And that sounds kind of like a big leap to go, all the way from mouse into human. But first in human clinical trials, epidemiology studies, you can see places for that kind of technology, that approach, that mutational assessment in those sort of situations where you've got populations that you want to be looking at. You may have some type of exposure that you think may be causing mutation. This is the assay that you could use for that directly in humans. And if you're interested in learning more about that, definitely contact us. I can send you some of the literature that we have published and see some specific case examples of how we use that technology in human samples.
Great. It looks like this next question is for Joe. Joe, can you describe a bit more about the automated gating?
Yeah, sure. So if anybody's done cytometry before, I'm sure that's kind of what motivated this question. But you're tasked with sort of isolating out the populations of cells. So to say these ones are positive, these ones are negative, and depending on how obvious that signal is, you may be on the hook to draw the line to say, here's where the negative stops and the positive begins. Most researchers are comfortable running controls to make that evaluation, but there's still some subjectivity to where those gates are placed. There's also a convenience in having it done automatically. So Miltenyi has a technology called an express mode, which I briefly mentioned, but basically it's a custom algorithm trained on data from your actual assay. So it can incorporate donor to donor variability, or really whatever the flex is in your assay itself. And it will physically draw the gates for you, spit out statistics, spit out a variety of things that maybe are manipulations of that statistic into something as a readout that you may use downstream. So super cool. There's a lot of different ones, but yeah, they fit nicely in this environment.
Awesome, thank you. How does the cell counting on the max quant work?
Yeah, this I was talking a little bit about the counting beads before. I'm not sure if I went into too much detail about that, but counting beads are a hard control that you would add to your sample to help qualify the number of cells in there. These were necessary on other cytometers because they don't qualify the volume going through very accurately. That's not a problem with the max quant because we operate on a syringe driven system. Basically think of it like a pipetter on board, and we have very precise understanding of how much volume is going through. And so that coupled with the event detection or count done by the cytometer itself allows you to work backwards and get a concentration super easily. And again, can spit out into like the actual data analysis software too. So it can save a few steps.
Wonderful. And it looks like we have time for one more question. Can the max quant be used for a validated assay?
Yeah, this is sort of a broad question, I would say, but it's an important designation to make that you can use these in a regulated setting. There are a number of things that you have to consider for that, but the short answer is yes, you can use this in there are max quants in validated or regulated settings, and we have a software package that's 21 CFR Part 11 compliant or helps support a compliant workflow. And that includes user management settings and analysis reports that are human readable and non-editable. So yeah, we have all the tools to help you get into a validated setting, and certainly we can help support that too if you're new to that environment.
Fantastic. Thank you. Thank you both. Do either of you have any final comments for our audience?
Jeff, you want to go first?
Yeah, I just want to say thanks for attending and listening to the presentation from both of our sides. Like Joe was mentioning earlier, we do have this really nice relationship back and forth between Litron and Miltenyi, and we've been trying to foster that for years, and it's served us really well on both sides. So we've got the methodology development, and in many cases, we're developing a lot of our assays for flow cytometry, and we have these instrumentation in-house. So we have several quants that we run our assays on when we're developing. So we immediately know how well it's going to work with these instruments. And that's just made a really nice sort of, back and forth when we're developing these assays. And I want to thank Miltenyi for the opportunity to come and talk about our assay as well.
Yeah, thanks a lot, Jeff. I fully agree. The connection between Miltenyi and Litron is a good one, and really helps foster new technologies or new assays as well. So yeah, thanks everybody for jumping on. If you should be able to find our contact information. Certainly Miltenyi's easy to find, Litron's easy to find. So if there are additional questions that you didn't get answered today, or something very specific to your particular challenges, just reach out. We're happy to answer questions. It's a big part of what we do. So yeah, thanks again for joining, and we appreciate your time.
Thank you again, Jeffrey and Joe, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.